In this figure, we're going to look at how protein structure dictates its function. Proteins have a very complex three-dimensional shape, which in many cases determines their function within the cell or sometimes when secreted outside of the cell. In this example, we're going to use a protein called calmodulin. This is an intracellular regulatory protein. In other words, it's a protein that becomes activated in response to cellular stimuli, and it transduces that stimulus into an intracellular response. Now, in the case of calmodulin, it is non-functional in the absence of relatively high concentrations of calcium. And we see it has this sort of arbitrary zigzag shape we've given it here. Now, when intracellular calcium levels rise, we have our calcium ions here, that calcium will become more likely to bind to particular sites on the calmodulin protein. And upon binding at those sites, what happens is we've changed the interaction between the folding elements of that protein. And in this case, we've sort of adjusted the shape here by having this one arm here sort of flip to the other side. Now, what that means in the case of this calmodulin molecule is we've generated an active site here, a pocket, if you will. And that pocket is adaptive to a particular intracellular enzyme that is, in this case, inactive in the absence of activated calmodulin. And so once our intracellular calcium levels rise, we intentionally change the conformation, the shape of our calmodulin molecule, which will now bind to our inactive enzyme, subsequently altering its structure and therefore function. So in the case of the inactive enzyme, we've now, through the binding of calmodulin, generated an active site that can now bind to and interact with substrate, making this enzyme active in the metabolic processes that it might be involved in. Now, this is what we would refer to as intentional modulation of structure and function of these intracellular proteins. We could also have unintentional consequences when the microenvironment of our cytoplasm changes. For instance, if the hydrogen ion concentration were to become too high, those hydrogen ions have the potential to interact with this protein and alter its function, possibly in ways that are dissimilar to the way calcium bound to and activated. It could be disabling to protein structure and function. And this is probably a good chunk of the rationale for such fine-tuning of the regulation of the intracellular microenvironment so that we maintain normal protein structure and function.